Hi, my name is Dominique. I'm a postdoc here. I'm in mineral sciences, but I'm actually a microbiologist by training. And thanks to this specific fellowship, I'm able to do interdisciplinary work here and combine those two quite disparate areas. So microbes are incredibly diverse. This is the only tree I'm going to show, and I'm only pointing out the things that I've circled include all animals, fungi, and plants, and everything else there we can't see. So that's the microbial world. They're everywhere, on and in us, and in the environment. And what's most interesting to me is that they drive a lot of elemental cycles. And this can be incredibly useful for bioremediation, where we try to use microbes to clean up metal pollution. So that quite scary looking photograph in the lower right corner is a stream in Pennsylvania that's contaminated with metals from an abandoned coal mine. So this is the area that I work in. And so manganese is an especially tricky metal to deal with. It stays soluble in water. So in the left part of that equation, it's manganese 2. And the way that we can get it out of water is through that oxidation reaction, where it gets transformed into a mineral form. Now, unfortunately, this reaction doesn't tend to happen spontaneously in the environment. It's mostly carried out by microbes. And so these microbes are already being used to try to clean up manganese contamination through these containment systems. And they're essentially pits filled with limestone that get colonized by these microbes. The contaminated water flows in, the microbes carry out that reaction, and the manganese basically crashes out of the water and accumulates in the bed where it's contained, and we can just remove it ourselves. So when these systems work well, they work quite well. The water coming out of the other end is fairly clean. Unfortunately, sometimes they don't work well at all, and we don't really know why that is. So the reason I'm interested in this is that it's essentially a microbiology problem, and so the goal is to increase the efficiency of these systems by looking at what's living in them. And so I want to know, well, how prevalent are these manganese oxidizing microbes? Are they all in there, so that indicated by the black cartoons there, or does the community look more like this, where the oxidizers are only a small proportion? And then, what are these other microbes doing? And could they be influencing how efficient these systems are? And so, for example, if we had a pure culture of a fungus that's pulling manganese out of water, so this is the reaction we want to encourage, you could see that if there are other microbes in there that are feeding the fungus, that could increase the amount of manganese that's removed. Whereas if you have other microbes in there that are eating the fungus or competing with it, far less manganese would be removed. So I want to look at everything that's in there, not just the oxidizers. And the way I do this, there's my advisor collecting manganese oxides from these treatment beds. I crushed them up and took out all the DNA, sequenced a short gene that's found in all organisms, and that gave me an idea of just how diverse these systems are and just exactly what the identity of these microbes is. And what did I find? A lot. Now, the details of this aren't important. What I really want to point out is just how busy this graph is. So the green and blue bars are two different manganese beds. The black is a clean, uncontaminated soil nearby. And along the bottom, the horizontal axis, are phylum-level classifications. So each of those groups actually includes a lot of diversity. These are very coarse groups, and yet there are a lot of them in here. And this is despite, metal levels in here that we would consider toxic. Now, my known manganese oxidizers, the one that we grow in the lab, account for less than 1% of the total. So there's a lot of other stuff in there that we actually have very little idea of what they're doing. And the surprise for me was this. There were a lot of algae in there. Now, you saw the picture of the sampling. That was very black manganese oxides. These aren't green treatment beds. And yet, lots of algae grow in there, very diverse. And so this is where my project took a bit of an unexpected turn. I thought it would be fun to try to grow some of these guys in the lab just to see what they're doing and to run some interaction experiments with the oxidizers. And so much to my advisor's dismay, I started filling her lab with flasks of green goo like that, covering all surfaces. And I found a lot of algae, and they will grow in pure culture in the lab. But the surprise was that if you can see in that flask, there's some black and brown kind of scum that started to form. And that turned out to be manganese oxide. So these algae are doing it themselves, never mind them feeding a fungus or a bacterium that's doing it. The algae, it turns out they can do it as well. And we had no idea that this was happening. And so if we had no idea that this quite important community component was directly oxidizing manganese, it begs the question, what are all those other guys doing? All those different phyla that we have in there. We could be missing quite an important part of the story. And so it just shows how important it is not always to look for what you're looking for, if you see what I mean. To step back and just take a shot in the dark, because you never know what you're going to find. And especially systems like this, where we're trying to influence environmental processes, we really need to take a broad step back and look at all the community members in these cases. So that's all I have. I'd be happy to take any questions. Yeah.
What are the algae doing with the manganese?
So the question is, what are the algae doing with the manganese? That's a very good question. It's the same, the question could also apply to the fungi and bacteria. What are they doing with it? We don't know. We don't know why they carry out this reaction. They don't seem to be gaining any energy from it. It's not like they're using the manganese as food. So my advisors are at Cara Santelli is working to show that it might actually be a side effect of their normal metabolism. So they might not be doing it on purpose. They might just be releasing some metabolites into their environment that have the effect of oxidizing manganese. Thanks.